concepts that we are going to deal with, ma. First is about ribosomes, which are non-membrane bound organelles, common in both prokaryotes and eukaryotes. Very good. Eukaryotes both in plant cell and animal cell. Tell me, what type of ribosomes do you see in a prokaryotic cell, ma?
70S.
Very good. 70S ribosomes. In eukaryotic cells? Very good. 70S and 80S. Okay. So, whichever ribosome it might be, it will have two subunits, yes or no? Okay. Larger subunit and smaller subunit. Smaller subunit will be like a cap over larger subunit, which is like a dome shaped structure. Okay. Larger subunit and smaller subunit. Okay. Let us say this has 70S ribosome. Okay. Larger subunit is what? Dome shaped structure. Dome like structure. In 80S ribosomes also it is common, smaller subunit over the larger subunit, like this. Okay, ma. Larger subunit is dome shaped structure again, and this is smaller subunit. Fine, right, ma? Ribosome consists of what? It has two subunits, okay. What are the components of ribosomes?
Proteins.
Only proteins? It consists of RNA and amino acids. Can you see here? So, these are nucleoprotein structures or nucleoprotein particles. Nucleoprotein particles. Okay. It has both RNA and amino acids. And then each ribosome will consist about 80 different types of proteins, 80 different types of proteins. Like if you see few subunits which are highlighted, if you have to consider 6 kingdom system of classification, who gave us 6 kingdom of classification? Then who gave us 5 kingdom system of classification? Who gave us 6 kingdom system of classification? Very good, Carl Woese. Carl Woese. 6 kingdom or 3 domain system of classification. There he said that there is one common ancestor for all the organisms. Okay, that common ancestor is known as progenote. From the common ancestor itself, we have 3 domains. What are the 3 domains? Archaea, bacteria, eukarya. As such, from these 3 domains, we have 6 kingdoms. Okay, what are the 6 kingdoms? Archaebacteria, Eubacteria, here Protista, fungi, plantae, animalia. Total 6 kingdoms, right? So, all of them having what in common? 16S rRNA in common. rRNA. Okay, like that we have the subunits over the ribosome. Here if you see the larger subunit of 70S ribosomes, you will be having 5S subunit and 23S. 16S subunit in the smaller subunit. Okay, these are different types of proteins, major proteins. Okay, here over the larger subunit you have 18S, 28S, 5.8S. Small correction, 18S in the smaller subunit. (28S), 5.8S, 5S subunits in the larger subunit region. These are the proteinaceous subunits that are present in the and smaller subunits. Very easy to remember. 18, 28, 5.8, 5. 5 is common in both the larger subunits. Here, 5.8. Smaller subunit has 18S. Nucleoprotein structure which consists of RNA and proteins. Fine, right? Clear about ribosomes? Now we have to know ribosomes present. We have already discussed about ribosomes in a prokaryotic cell. Where are they present? Attaching to the plasma membrane and in the cytoplasm. Both are 70S ribosomes only. Ribosomes major synthesis is what? Ribosomes plasm of prokaryotic cell, they are involved in protein synthesis. Those which are attached to plasma membrane of prokaryotic cell, they are involved in translocation of proteins. Clear, right? Translocation of proteins. So you are clear with the prokaryotic cell ribosomes. Now we will discuss with eukaryotic cell ribosome, plant cell. Okay. Consider okay. Ribosomes. Inside the matrix of mitochondria, what do you have? 70S ribosomes. Okay. In the cytoplasm, you have capacity. Same the case with animal cell. Of ribosomes, their synthesis, and what are the subunits that are. In eukaryotes, over the membrane, do you not find any ribosomes? There is no certain exception. Completely absent in the plasma membrane region. Based on the functions and translocations, they will be transpanning. Okay, but when you are learning the fluid mosaic model, nowhere we have learned the ribosomes. Clear, right? Prokaryotic cells, as they are unicellular, protein translocation is mandatory for them. Okay, that is why few ribosomes will be attached to the plasma membrane in prokaryotic cell. Good doubt. Any other? Untai. That we will learn when we are learning about the nucleus. We will learn. Any other doubt? Till here it is clear. No? We will learn about one more non-membrane bound organelle. Okay, these are centrioles, present only in the animal cells. Only in so here if you see, you consider this as a cell, single cell, first you have to know what are the major uses of the centrioles. Usually most of the students will get confused between the centriole structure, cilia and flagella. What is this and what is that? What is the duty of this? What is the duty of that? First you remember about centrioles. Centrioles are the structures found only in the animal cell, and how they are present? A centrosome is present which doesn't have any membrane. Inside this centrosome you have two perpendicular structures like this, two structures perpendicular to each other. Okay, these cylindrical structures which are perpendicular to each other are known as centrioles. So in a single centrosome, how many centrioles are present now? Two centrioles are present. Okay, they are perpendicular to each other. As a single centrosome consists of two centrioles, this is known as diplosome. Fine, right? This is known as diplosome. Clear? Any doubts anywhere? Now you look at the activities of the centriole. Centriole will help in the release of spindle fibers, in radiating out the spindle fibers. If you have to learn about cell cycle, there we have learned during S phase, DNA duplication happens inside the nucleus, centriole duplication happens in the cytoplasm. Okay, so these centrioles after duplicating, they will move out to the opposite poles like this in the form of asters. Remember, these asters will radiate out the spindle fibers. These spindle fibers will be attached to the kinetochores of the chromosome. Okay, at the center you have centromere. Don't get confused between these two terms. This is centromere, this is centrosome. Okay, centromere which is at the center, this centromere will be having two disc-shaped structures called as kinetochores. To this kinetochores, the spindle fibers will be attached. First, these are radiated from what? Asters. Asters are nothing but centrioles which have moved to the opposite poles during prophase. Understood, ma? Clear? Now you have understood the story behind. This is the major duty of centrioles. Second duty, that over a cell, eukaryotic cell motile structure also flagella is present, yes or no? For that, first it will be originating, flagella structure will be originating from cell membrane, and then it will be extended from the cell wall. Yes or no? It will be originating from cell membrane and extended from cell wall. And this forms out the basal body of the flagella. Okay, we have three components of flagella, right? Basal body.
What is the properties of?
Very good. Okay, basal body, hook and filament. Basal body originating from cell membrane, extending from cell wall. Here it has some rings. These rings are LPSM rings. These rings, they are attached to the cell via centrioles. That is the reason the basal body of flagella composition will be 9-0 microtubular arrangement. Understood? These are the roles of the centrioles which are present inside the which cell? Animal cell only. Protozoan cell is an animal cell or not? Protozoa. Animal cell only. Cyanobacteria? Prokaryotic cell. You have to know the difference. Why? Because protozoans have a membrane bound nucleus, they don't have a membrane bound nucleus. Simple. Simple characterization between prokaryotic and eukaryotic cell. Whenever you are learning about unicellular organisms, all the examples of your NCERT, you have to be clear about what are prokaryotic, what are eukaryotic. Goniolax is prokaryotic cell or eukaryotic? Goniolax. Trichodesmium erythraeum. Trichodesmium is the example of? Take down. Question for you. If you identify, you will say me in the next class at least. Goniolax and trichodesmium. Write it. Separate note. Trichodesmium. Trichodesmium erythraeum. Good. Eubacteria. Under cyanobacteria you have learned, right? Both of them cause the same activity, that is red tides over the Red Sea. Okay. They cause red tides of, just a second. Is the voice clear now? Okay. Red tides over the Red Sea, they will cause, we have learned two examples, one under cyanobacteria, one under dinoflagellates. One is prokaryote and the other is eukaryote. Because this comes under protista, that comes under monera. Getting the point or not, everybody? So now about centrioles it is clear, right? So when you have to take the central part of the centromere, if you dissect and see at the central part how they are arranged. At the center there is a proteinaceous hub. There is no microtubule at the center. There is what? A proteinaceous hub at the center. From this proteinaceous hub, there are 9 radial spokes that are radiating out. How many, ma? 9 radial spokes. To each radial spoke, there are 3 peripheral triplets. 3 peripheral triplets named as A, B, C. Okay. C microtubule of one radial spoke is connected to A microtubule of the next radial spoke. This is called as CA connections by the protein nexin. Nexin, dynein, these are the protein connections that are present in the centrioles. Okay. So at the center there is proteinaceous hub from which there are 9 radial spokes that are originating. Each radial spoke will have how many triplets? 3. Okay. 3 microtubules, which is a single triplet. C microtubule of one radial spoke is attached to A microtubule of another radial spoke. Okay. Clear? Just a second, we'll fix it out. Is it fine now? Can you hear me? Voice is clear, yeah. So now about centriole, it has one proteinaceous hub at the center towards which 9 radial spokes are present, each radial spoke 3 microtubules. So on total in a single centriole, how many microtubules are present, ma? 27. Very good. That is 9 into
See, when we talk about
microtubules. And you are also clear with the functions of centrioles. The primary function is formation of spindle apparatus. Okay, formation of spindle apparatus. Second is formation of the basal bodies as well, of cilia and flagella. They form the basal bodies of cilia and flagella. Clear with both the functions of centrioles? In a single centrosome, how many centrioles are present, ma?
2.
How they are arranged? Perpendicular to each other. That's it. Clear, right? Centriole will have centroplasm. That's it. They don't have any membrane. Okay. Next. About cilia and flagella. Unlike the centrioles which you had, where you have seen at the center there is a single proteinaceous hub, but here you can see at the center there are doublets, microtubular doublets. Okay, that is there are no alphabets to it. At the center you have a central doublet which is enclosed by a central sheath. Central microtubules, how many you have? 2 central microtubules which are surrounded by a central sheath. Attaching to this central sheath, you have 9 radial spokes. Okay. To each radial spoke here, unlike centrioles, here you have a doublet. Okay. Here you have what, ma? Doublet, which is AB. Peripheral microtubules which are AB. Okay. B microtubule is adjoined to A. Fine, right? These are peripheral microtubular connections. Here you will see BA connection, there it is CA connection. So on total how many microtubules are present? Number of microtubules. Central microtubules are 2 plus 9 radial spokes. Each radial spoke is having 2 microtubules. So 2 plus 18, on total it is 20 microtubules. Okay, and what is the protein that is present here? Tubulin. This is about cilia and flagella, which is what? Eukaryotic flagella and cilia. Cilia is the motile structure of only eukaryotic cells, not prokaryotic cell. Remember that. Only motile structure of prokaryotic cell is flagella. Okay, only motile structure of prokaryotic cell is flagella. Eukaryotic cell you will have cilia and flagella. With what microtubular arrangement? 9 plus 2. Okay, how the flagella will move? Not like this. It will be rotating, flapping out, and these microtubules will be rotating like this. That is how the flagella movement takes place. Fine, right? Same the case with centrioles when they are moving to the poles. Understood about cilia, flagella, centriole? Any doubts anywhere? Go through your NCERT, just 2 minutes I will give you the time. About cilia, flagella and centriole. 2 paragraphs you have, right? In my presence, right away you read. Quickly, if you are having any doubt, you can ask me. Good morning. The voice is clear only. If you are done with that, just let me know, ma. We will move on to the cytoskeleton structure. Done? Any doubts anywhere? Clear right now? Yeah. Now look at the cytoskeleton. The name itself is saying cyto is cell, skeleton is structure, which gives proper structure to the cell, helps in both intracellular and extracellular motility of the cell, for which we have 3 major proteinaceous structures. Those are microtubules, microfilaments and intermediary filaments. Microtubules will have tubulin protein. Okay, these are unbranched hollow tubular structures. Inside this unbranched hollow tubular cylindrical structures, each tubulin protein will have 13 protofilaments at the center of this. 13 protofilaments. Fine, right? What are the major functions of microtubules? Constitute spindle fibers. Okay, microtubules are present in spindle fibers, chromosome fibers and basal bodies, flagella and cilia. In all these bodies, what are the major components? Microtubules are present. Fine, right? In spindle fibers, chromosome fibers, basal bodies, flagella and cilia. They also function as cytoskeleton, giving proper structure to the cell, and an intracellular transport. Why intracellular transport is required? Yesterday we have learnt endomembrane system, where the targets from ER should be passed to Golgi. From Golgi, targets of vesicles should be passed to the lysosomes, vacuoles, and to the plasma membrane, like that. To many intracellular targets, how they are transported via microtubules. They control the shape of the wall-less cells like mycoplasma, okay, and they help in movement of nuclei during cell division, that is during karyokinesis, and it also helps in the formation of cell plate. We have learnt how a cell plate is formed, and it also helps in cell movement, okay. So microtubules, major function, many at times asked questions. You have to be clear in your mind where microtubules are helpful, okay. They are helpful in, they consist in spindle fibers, fibers, basal bodies, flagella, cilia. Functions as cytoskeleton, intracellular transport, control shape of wall-less cells, movement of nuclei, that is in karyokinesis, cell plate formation, and in the cell movement as well. These are the major functions of microtubules. What is the protein that is present here? Tubulin protein. They are branched or unbranched? Unbranched cylindrical rod-like structures. At the center you have 13 protofilaments. Clear? Any doubts anywhere, ma? Yeah. Now you see microfilaments. Microfilaments will have two proteins, globular protein actin and one more protein, that is myosin. Okay, it has two proteins, actin and myosin. Actin, globular protein, and filamentous protein. There are narrow cylindrical rods or protein-like structures. They mainly help in cytoplasmic streaming. Any components to stream in the cytoplasm, microfilaments will be used. Formation of fibrils. These are muscle fibrils. Whenever you are learning about muscle contraction and relaxation, you will be learning about that. Next, formation and movement of microvilli, which is present in the small intestine. Formation and retraction of pseudopodia, which is false feet of amoeba. Okay, in the movement of that, endocytosis and exocytosis. Okay, formation of spindle apparatus. They are not as the constituents of spindle apparatus, but they help in formation of spindle apparatus. Movement of cellular components. So whenever there is an option that is given with respect to cytoplasmic streaming, movement, pseudopodia movement, and formation of spindle apparatus, not constituents but formation of spindle apparatus, and movement of cellular components, then you have to choose what, ma? Microfilaments. And what are the proteins it consists of? Actin and myosin. And it is also unbranched structure only. Unbranched is tubular structure, not branched structure. This is also unbranched. Next is about intermediary filaments. It has solid unbranched filaments. They are formed of variety of proteins. It consists of many of proteins which are of acidic in nature, ma. They are of acidic in nature. Major functions include formation of nuclear matrix, supports membrane, supporting array of cytoplasm, contractile units, and mechanical strength to the nervous tissue. Fine, right? These are the major functions. Don't look at the theory of this. Just remember what is the role of different proteinaceous filaments. That's it. Okay. Cytoskeleton consists of three major proteins: microtubules, microfilaments, intermediary filaments. What is the role of each proteinaceous filament? What are the major functions of it? That's it. Okay. Clear right now, ma? I'll give you 1 minute of time. Just go through the table. If you remember this table, you can answer any question from cytoskeleton. Done right, ma? You can copy it later. Just look at the functions. Just remember that. Look at it and remember. Everything will be saved. You can copy it later, okay? Now we will move on to double membrane bound organelles. What are the double membrane bound organelles, ma? Nucleus. First we will look into mitochondria structure. Mitochondria can be of cylindrical or sausage shape, that you know. Okay, it can be cylindrical or sausage shaped, whose diameter can be from 0.2 to 1 micrometer in diameter, and whose length can be from 0.4 to 1 micrometer in 1 to 4.1 micrometer in diameter. Okay, sorry, in length and in diameter. This go through that. This will be from 0.2 to 1 micrometer, and this length will be from 1 to 4.1. What shape it is? Cylindrical or sausage shaped. For your basic understanding, let me draw a mitochondria, a cartoon structure of mitochondria. Okay, so the inner mitochondrial membrane, which is also known as cristae, right ma? Inner mitochondrial membrane or cristae. It has many enfoldings, right? This is separating the mitochondria into mitochondrial matrix and outer compartment. It is giving out two compartments here. One is inner compartment and the other is outer compartment. Outer compartment is known as intermembrane space. Okay. Inner compartment is known as mitochondrial matrix, and this is outer mitochondrial membrane. So you have four major components. Firstly, you have to remember the inner mitochondrial membrane is enfolded in the form of cristae, and this is dividing the mitochondria into two compartments. One is inner compartment and the other is outer compartment. Inner compartment is addressed as mitochondrial matrix. Outer compartment is known as intermembrane space, right ma? So here outer mitochondrial membrane consists of lipids, proteins, and it is freely permeable. Inner mitochondrial membrane also consists of lipids and proteins, but it is selectively permeable. Okay, it is selectively permeable, and it consists of special types of lipids known as cardiolipins. How much percent of proteins and lipids are present here? Protein percentage is 6%, lipid is 40%. Here if you see, protein percent is 80%, lipid is just 20%, and that is also special types of lipids, cardiolipins. Fine? Okay. Simple and easy. Look at here once everybody. Inner mitochondrial membrane or cristae into two compartments. Inner matrix, outer intermembrane space. Here protein and lipid composition. Inner mitochondrial membrane, protein and lipid composition, and what are the different types of lipids that are present. That's it. Now we look into the components, which are very important. You know, mitochondria which is present in a cell is known as what? Powerhouse of the cell. Why is it addressed so? Because mitochondria is helpful in the synthesis of what? ATP. ATP is addressed as cellular energy.
Okay. So they are given. Okay, this is little different.
First form of energy where the cell is used for its metabolism. Okay, it cannot use a complex food material like NADH and FADH. Okay, always our mind or anything will perceive the simplest forms only. So the simplest form is providing, it is known as powerhouse of the cell. How many number of mitochondria are present in a cell? That is requirement, that is based on the energy requirement of the cell. Number of mitochondria will vary. Okay, and here you have to recall few functions. What are the components of each structure? You have to know. You remember all the protein channels electron carriers whichever are required for the synthesis of energy or mitochondrial activity, they are present in the inner mitochondrial membrane only. Okay. All the electron carriers, protein channels required for the synthesis of energy are present where? Inside the inner mitochondrial membrane itself. And in the matrix of mitochondria, you will have a double stranded circular nucleus, double stranded circular DNA, 70S ribosomes, RNA and proteins. What RNA are present, ma? 70S ribosomes and RNA, whichever are required for protein synthesis. Okay. 70S ribosomes, RNA, proteins, double stranded circular DNA, they are present in mitochondrial matrix. Whichever electron carriers, protein channels, ATP synthase complexes, they are present in inner mitochondrial membrane. Fine. If you are clear with this, once we will have a gist of aerobic respiration, like where the activities are happening. You are clear with this, right? We can move. Yeah. Here you look into, ma. First, if I have to consider this as a cell, fine. First, glycolysis occurs, yes, right? Whenever food enters into the cell, okay, let us suppose glucose has entered into the cell. Now from the food what we will get? Energy. Food gets oxidized and we will get the energy. Energy production role is what? Mitochondria's role is to produce the energy. So when glucose enters into the cytoplasm of the cell, first it gets break down to pyruvate. Very good. 2 molecules of pyruvate. So this first step is what, ma? About energetics, about process, we will discuss in the respiration chapter, but here a gist for you where activities are happening. Okay. This pyruvate will directly land into which region of mitochondria? Can anybody tell me? First you mark out the regions. This is what? Inner mitochondrial membrane or cristae. This is what, ma?
Outer mitochondrial membrane.
What is this? This region?
Matrix.
Okay. So remember, first this pyruvate will land into the mitochondrial matrix region. Immediate function that happens is second step, oxidative decarboxylation. Oxidative decarboxylation. And the second step is Krebs cycle, and then it will be synthesizing acetyl-CoA, right? Now you got it. This acetyl-CoA is the starter of TCA cycle. Now you got it, I know. Both of them will happen in matrix of mitochondria only. So at the end of TCA cycle, if you have to remember, you know that it produces the high amount of energy. NADH is produced, FADH2 is produced, okay, only some amount of ATP is present, right? Now this NADH, FADH2, it has to dehydrogenate, okay, they have to be oxidized and the energy have to be released in the form of ATP. That activity, which is the last activity, is done in the intermembrane space with the complexes that are attached to inner mitochondrial membrane. Inner mitochondrial membrane. 4 complexes are present which will activate electron transport system, which is the fourth and final step of aerobic respiration, that is electron transport system. All the complexes related to electron transport system are present in the inner mitochondrial membrane facing towards intermembrane space. Any doubts anywhere? Clear, right?
Yes.
Clear? You are clear with the components of mitochondria and the functions of mitochondria. Any doubts anywhere, ma? Shall we move on to plastids? Yeah. Take 2 minutes, just fix everything in your mind, and if you are having any doubts regarding mitochondria, you can ask me.
What carries pyruvate into matrix?
Pyruvate into matrix. The molecules will just land here because their functions are here. Okay, there are many receptors, protein channels that are present, not required for you. Okay, everything is being transported by proteins only in the form of receptors. It will be accepted in the form of receptor, transported in the form of protein channels, not required for you.
In mature RBCs, mitochondria is absent. Mature RBCs, mitochondria is absent.
Mature RBC. Golgi bodies are absent,
Mitochondria.
is also absent.
Then what provides energy?
What is the role of RBC?
Carrying oxygen.
Its role is always to transport. Okay, and it gets the energy in the form of transport. What is the major pigment that is present in the RBC?
Hemoglobin.
It binds more to.
Oxygen.
That is how the transport take place. There is no rail, yeah, no this. Done? Yeah. Now about plastids. We have 3 types of plastids. What are they? Plastids are again double membrane bound. Yes or no, ma? Plastids are again double membrane bound. We have 3 types of plastids.
Into Y minus.
First is chloroplast. Next is chromoplast. We will learn the structure and function of chloroplast in detail first. Listen, look at the structure. It is also having 2 membranes. Outer membrane is permeable, inner membrane is selectively permeable. Okay, so in between these two, in between outer membrane, outer plastid membrane and inner membrane of this, there is some space that is present which is called as periplastidial space. Okay, and the matrix of chloroplast consists of what? Stroma. What is the matrix of chloroplast? Fluid of chloroplast? Stroma. And here you have stalk of sac-like structures. Single membranous sac-like structures are known as thylakoids. You have a membrane which is also known as third membrane system of chloroplast, which is thylakoid membrane. Thylakoid membrane is also addressed as what, ma? Thylakoid lamellae. Thylakoid membrane is also known as thylakoid lamellae or the third membrane system of the chloroplast. At the center you have what? Thylakoid lumen. Fine, right? So this is the membranous sac-like structure. Stalk of membranous sac-like structures are known as grana. One grana is adjoined or connected to the other grana by stroma lamellae. One grana connected to other grana by stroma lamellae. Clear, right? Double membrane, space in between 2 membranes, periplastidial space. Matrix of the chloroplast is stroma. Inside stroma you have membrane sac-like structures called as thylakoids. At the center you have thylakoid lumen. Okay, stalk of the sac-like structures grana. One grana to other grana by they are just placed like coins, pile of coins. Inside stroma of the chloroplast, that is fluid filled space, what are present? Double stranded circular DNA again. inside stroma region. Double stranded circular DNA is present. 70S ribosomes, RNA and proteins. Same constitution like how it is present in mitochondrial matrix. What is the role of double stranded circular DNA which is present both in chloroplast and mitochondria? What is the role of this double stranded circular DNA? Of course, chloroplast, mitochondria, they share the traits of prokaryotic cell and they multiply by fission only, binary fission, and they have 70S ribosomes like how a prokaryotic cell consists of. Okay, what is the role of this? Because it has its own DNA, it is addressed as semi-autonomous cell organelles, both chloroplast and mitochondria. Semi-autonomous cell organelles or self-duplicating cell organelles. Fine, clear? And they take care of their own protein synthesis. Fine? Double-stranded circular DNA, 70S ribosomes, RNA and proteins, these are the constituents of stroma. You are clear with the structural details of chloroplast? Okay. Thylakoid lamellae, stroma lamellae. We can also call it as stroma lamellae. There is a membrane above the stroma also, no? That is stroma lamellae, and this is thylakoid lamellae, which is nothing but membrane only. Any other? Yeah, now you see here. You know, inside a chloroplast, if you know the details of the functional details, like where what role is happening both inside mitochondria and chloroplast, it will be easy when you are learning plant physiology. That is the reason I am highlighting you the details. Inside the chloroplast, two activities majorly happens in photosynthesis. What are they? Light, which are addressed as light dependent reactions. Light independent reactions. Calling light independent reactions as dark reactions is a misnomer because it is not dependent on light directly. It takes place both in light and dark. Okay. So here there are three major events of light dependent reactions or light reactions. First is absorption of light in the form of photons. Second is photolysis or hydrolysis of water. Third is formation of assimilatory power.
No, the graphs. Okay.
Includes both ATP fine. Where is the absorption of light in the form of photon takes place? We have learnt thylakoid has a membrane which is known as thylakoid membrane or thylakoid lamellae. It consists of photosystems. PS2, PS1. They have light harvesting complexes. They will trap the light. Photolysis of water takes place in the inner membrane of thylakoid. Here there is a complex that is present, oxygen evolving complex. Okay. So here this happens in inner thylakoid membrane. This happens in thylakoid membrane. Locations are very important, sites of reactions. Absorption of light in the form of photons in thylakoid membrane, photolysis, inner side of the thylakoid membrane, formation of assimilatory power in the stroma. Light independent reaction, only one reaction happens, only one role, that is synthesis or formation of glucose.
Okay.
Glucose formation.
Able to see, or you want to see?
Takes place where inside stroma or thylakoid. That's it. You are clear with this, right? As well as this. Chloroplast, mitochondria is clear to you. Now look at the leucoplasts. What are the leucoplasts? Those plastids which are colorless and those which store the food materials. Examples include what more? What are different types of leucoplasts? Based on the food material it is storing, we have three types of leucoplast. First is amyloplast. Second is elioplast. Third is aleuroplast. Aleuroplast stores, example is maize. Elioplast stores oils and fats. Amyloplast carbs or carbohydrates, starch.
What is the AD, that's all. Okay. So,
Okay. Chromoplast will contain different range of pigments like carotenoids, xanthophylls, anthocyanins. Okay. They exhibit different colors based on different pigments. You can see the leaves which are of different colors other than green. Few leaves with green and other colors which are known as variegated leaves. Okay. That is because of what? Chromoplasts which are present in the mesophyll cells. These are the organelles which are present in the mesophyll cells that emit different colors on the leaves. You can note down the pigments. Done, right? Clear with plastids? Shall we move on to nucleus? What happened? Why you are required? Fine, right? This is clear. Yeah. Next is about nucleus. Nucleus has four components, ma. Four components. One is nuclear envelope. Second, nuclear pores. And third, nucleoplasm. Fourth, nucleolus. Fine, right? Nuclear envelope, nuclear pores, nucleoplasm and nucleolus. Nuclear envelope is double membrane bound. In between you have pores which help in translocation of materials into the nucleus and out of the nucleus. The space inside the nucleus is addressed as nucleoplasm, and inside the nucleus there is a non-membrane structure which is addressed as nucleolus. Okay. RNA synthesis. We have three types of RNA in a eukaryotic cell. RNA synthesis will happen inside the nucleus. For example, if I have to suppose this as nucleoplasm and this as nucleolus. Inside nucleoplasm, tRNA and mRNA is synthesized. Fine, right? Inside nucleolus, rRNA is synthesized. rRNA is synthesized by RNA polymerase 1. mRNA is synthesized by RNA polymerase 2. Very good. tRNA by many a times asked question in connection with molecular bases. Okay, remember that synthesis. And here inside the nucleoplasm, the chromatin network which is present, it is present in the form of thread-like structure only, completely in the form of chromatin network itself. Only at the time of karyokinesis it starts condensing. Condensation happens at the beginning of karyokinesis in the time of prophase. Rest all the phases in the interphase might be, after telophase might be, it will be in what? Chromatin structure. Now we will look into the components of chromosome also. Once if this is clear, I will tell you. And I already told you, nucleus number may vary with different cells. If you have to consider sieve tube cells, mature RBC cells, there is no nucleus. If you have to consider opalina cells, multinucleated, okay. If you have to consider paramecium, okay. Food vacuole over here, and here it has two nucleus, macronucleus and micronucleus. Micronucleus will help in the reproduction, macronucleus helps in metabolism, conducting metabolism of metabolic activities of paramecium. Okay, any doubts anywhere? Clear, right? Yeah, this is about nucleus. And then, yeah, chromosome you see clearly. Be here everybody.
What is this?
Yeah, at the center you have centromere. This is centromere. Adjoining to the centromere you have two disc-shaped structures. These disc-shaped structures are kinetochores. To this kinetochores only what are present? Spindle fibers are attached. From this centromere at the center, you can see chromatids are connected. Okay, you can see how many chromatids are here? Two chromatids, and these are arms of the chromatids. Okay, two chromatids, and these are two arms of the chromatids. Based on the shape differences of the chromatids, we have two types. We have four different shapes of the chromatids. One thing you have to be clear, at which phase you can see clear structure of the chromosome. No. You can see clear structure of the chromosome. So you are clear with the structure, right? Chromosome structure. What are the components of a nucleus? What are present? DNA is present, yes or no? DNA is negatively charged or positively charged? Negatively charged. Because it has phosphate group which is phosphoric acid. This is negatively charged. So to pack the DNA in this short space of nucleus, it will be adjoined with the positively charged histone proteins. Okay, in the higher level of condensation of DNA, there is also requirement of non-histone proteins. Okay, along with this what is present? RNA is also present. So we have DNA, histone proteins, non-histone proteins, RNA in the chromatin which is present in the nucleus. Fine? And this is the structure of the chromosome. Yeah, ribosomes are also present. And then shapes of chromosomes, ATS. Shapes of the chromosomes. We have four different shapes of the chromosomes. Shapes of chromosomes are clear in which phase? Clearly visualized in which phase? Metaphase. No, anaphase. Structure is clearly visible under microscope during metaphase. Shapes of the chromosome during anaphase. It is because all the chromosomes get thick and short when they are adjoined to the equatorial plate during metaphase, right? At the equatorial plate, all chromosomes will be thick and short. They will be adjoining to what? Equatorial plate, like this. To the kinetochore, spindle fibers will be attached. Okay, but if you consider the anaphase, centromere splitting will happen, right? After centromere splitting, the chromatids will separate and they will move in different shapes. So shapes of the chromosomes will be clear in anaphase. That's it, as simple as it is, right? Here you see, first is metacentric chromosome. About shapes of the chromosomes, many times they have asked a question, listen carefully. Metacentric. When metacentric chromosome undergoes centromere splitting, how chromosomes will be moved in what shape? In V shape, because they have two equal arms of the chromatids. This is metacentric chromosome. Okay, one long arm of the chromatid and one short arm, which is submetacentric. Submetacentric, which is of what shape, ma? L shape. Okay, and this is acrocentric. One very short arm, one very long arm. They will be moving in like this, J shape. Okay, and then telocentric. One extremely short, one extremely long, which is I shape. Fine, clear? So we have metacentric, after splitting it moves in V shape, submetacentric L shape, acrocentric J shape, telocentric I shape.
Means what?
One long arm and one short arm.
This
Both in submetacentric and acrocentric is addressed as Q arm. Short arm is addressed as P arm. Long is Q, short is P. Fine. And in few chromosomes, there is a secondary constriction. At the center here we have centromere. Centromere is the primary constriction of the chromosome. There are the secondary constriction. Above the secondary constriction there are some DNA sequences. These are known as satellite sequences. Satellite will have most of the repetitive sequences of the DNA. In human nucleus, if you consider, there are 5 satellite chromosomes. 5 above secondary constriction. Okay, not all chromosomes will have secondary constrictions, only few will have. Above the secondary constriction, whatever sequences are there, you will be calling it as what? Satellites. Satellite chromosome. Understood about different shapes of the chromosome? So, a quick recap. What did we deal today? First about starting non-membrane bound organelles, about ribosomes, different types, where they are present. Followed by centrioles, followed by cilia and flagella, and then cytoskeleton, and then mitochondria, followed by chloroplast, and then nucleus. Yeah, in plastids we have learnt chloro, chromo, leucoplast, and then about nucleus, then chromosome structure, and then different shapes of chromosome. This ends the complete chapter. Okay, quickly let us look at our memory, a small memory test. Let us solve few objectives of this. Yeah, this you have solved yesterday, no ma? Yeah, this one. Option 2? Yes, okay, we'll see. Cilium or flagellum, which is a singular form, contains an outer ring of 9 doublet microtubules surrounding 2 singlet microtubules. Cilia are smaller, which work like oars, causing movement of either cells or surrounding fluid. They will be swimming like this, right? If you can remember the structure of paramecium, which has thousands of cilia. One more point to highlight, ma. Amongst flagella and cilia, which is serving the purpose of fast locomotory activity, means your answer should be cilia. Next, flagella are comparatively longer, responsible for cell movement. Cilia and flagellum are covered by plasma membrane. Good. Good, good. Yeah, this one you have already done this, right? Yeah, this one. First question, assertion reason. What is the option?
Based on this.
A, how many? Raise your hand. B, rest, no option. Okay, listen. Ribosomes in prokaryotic cell will have 70S sites. The S in ribosome stands for Swedberg unit and measure of sedimentation rate during centrifugation. See, in reason he is explaining about S, no? In reason he is explaining about 70S type, right? So assertion and reason are two. Reason is the correct explanation of assertion. See, any time statements are given which are unconnected to both of them, but both are correct. In that case only you have to choose option B. In rest of the cases where reason is explaining the assertion, you have to choose option 1. Next question. Which of the following is or are not the functions of Golgi apparatus? Sorting and packaging of proteins is nothing but every protein is being targeted to different targets, right? It is sorting and packaging. Lipid breakdown. That is not the activity of Golgi bodies. Next. Golgi bodies, smooth endoplasmic reticulum, ribosomes, lysosomes, first question. Mitochondria contains single circular DNA and 70S ribosomes. It has single circular DNA and 70S ribosomes. You have to tell me, I cannot help you. Listen to the statement properly, look at it properly. See, how many of you are saying one is incorrect? Why is it incorrect? He is saying single circular DNA. Yes, every mitochondria will have only single circular DNA. He didn't mention about single stranded or double stranded. Did he say single stranded circular DNA? No, no. Then it will be wrong. He is saying single circular DNA. Yes, every mitochondria will have single circular DNA. He didn't mention about strands. Look at the statement properly. Understood? Next question, assertion and reason. Centrosomes are found in animal cells. In few plant cells they are present. They are absent in all plant cells. Plant cells perform mitosis without formation of spindle fibers. No, spindle fibers will be formed, but they are addressed as microtubular organizing complex. Okay, that we will deal in cell cycle and cell division. This question.
సెంట్రోజోమ్లో సెంట్రియోల్స్ ఉంటాయి కదా మేడం, ప్లాంట్ సెల్లో ఉండవు.
ఉండవు. They will have microtubular organizing complex which will help in spindle fiber formation, that I will tell you at the time of cytokinesis in cell cycle and cell division. This question you will answer there bright, but now you remember centrosomes are found in animal cells and they are absent completely in plant cells. Assertion is wrong. Plant cells perform mitosis via spindle fibers, but they are formed through microtubular organizing complex. So here you can go with A is false and R is true. Next. Simple, see and answer, applied. Be louder and say what is the right answer. Oh my god. Pillai in bacteria help in conjugation, yes or no? Yes. Plant cells do not have lysosomes. Is that statement true? Wrong. They do have lysosomes. ATS ribosomes are found in cytoplasm of eukaryotic cell. Yes? Yes. Prokaryotic cells have double membrane bound organelles. Wrong. Okay. Okay, cool. You can go with 1 and 3 are the correct answers. If I have to give, now you tell me. B. Assertion, what did he give? In amoeba, contractile vacuoles help in osmosis. We discussed yesterday. Vacuoles in protists function primarily for digestion. Food vacuoles which are present in paramecium helps in digestion, but he didn't mention it right away. We have 3 different types of vacuoles. So here A is true, R is false. Next, match the following type of question. Options will be given in the next slide. Last question. Both A and R are true. Is it explaining or not? Yes, ma'am. Next. Which of the following are true for chloroplasts? They have double membrane bound organelles. Chloroplast itself is an organelle. They have 80S ribosomes. No. Thylakoids are arranged in grana. True. They possess circular DNA. He didn't mention whether it is single or double strand. So the more evident option is C. Assertion and reason. Ribosomes are called as protein factories. True. They are involved in DNA replication. No, they are involved in protein synthesis. Ribosomes are composed of RNA and proteins, and they exist freely or attached to. Next. Very good. Which is option? A. Next. Eukaryotic flagella exhibit 9+2 arrangement of microtubules. The axoneme is nothing but the central part, how we are seeing that arrangement. Of cilian flagella contains 9 peripheral doublets and 2 central singles. Next. Centrioles, axoneme, nuclear pore, nucleolus. Next. D. Centrosomes organize spindle fibers during cell division in animal cells. True. Centrosomes, inside that centrioles are only present, right? Nucleolus is membrane bound. Wrong. Nuclear pores allow bidirectional transport of RNA and proteins. True. Cilia and flagella, yes, they will allow bidirectional transport of RNA and proteins for post-transcriptional events and translational events. Cilia and flagella are covered with plasmamembrane. Here you take incorrect. Then the answer will be B. Next question, ma. Paramecium we have discussed. Chlamydomonas will have single nucleus. Sieve tube cells nucleus is absent. Erythrocyte also nucleus is. Next. Centriole microtubule 9+0, while axonemes 9+2. This question. A, both A and R are true and R is correct. Metacentric chromosomes have equal arms. Yes. The centromere is located close to one end. Wrong. Centromere is located at the center. Last questions. on the shapes of chromosomes. Centromere location he is asking. Centromere location. Metacentric, centromere at the center. Submetacentric, acrocentric, telocentric. What is the option? Done. That's it. So with this the complete chapter is done. We'll move on to cell cycle in the next class.